to introduce Dr. Jose Zambrano with EPA's National Exposure Research Laboratory, who will briefly introduce the topic and our presenter for today.
Thanks, Lisa, and can you hear me okay?
Yes.
Okay, great. Well, greetings to everyone. Thank you for joining today's webinar. I am a Senior Science Advisor at the National Exposure Research Laboratory, which is one of ORD's 3 national labs with centers for research around the country, and really excited to be able to introduce today's topic. The lab actually is the Center for Improving Measurements and Methods and Models to Assess and Predict Exposures of Humans and Ecosystems to Pollutants and Other Conditions in Air, Water, Soil, and Food. And we are honored today to have Dr. Jay Garland, who's the Director of our Systems Exposure Division, one of the 3 science pieces of NERL, who to give today's seminar. Dr. Garland joined us in 2011. He has a PhD in environmental science and has spent over 20 years working on NASA's efforts to develop closed bioregenerative life support systems for extending human spaceflight. And we are fortunate that he has come to EPA and has been working with us on addressing research gaps to advance decentralized water reuse and inform risks associated with antimicrobial resistance in the water cycle. Dr. Garland has a wealth of experience internationally as well, and I'm honored to turn it over to him to talk about non-potable water reuse. Dr. Garland?
One quick thing, if I may, we are going to mute.
The leader has muted your line. To unmute your line, press #6.
All right, I think I've unmuted myself, so hopefully you can all hear me. This is Jay Garland. It's a pleasure to be able to talk with you today, because I think, as you'll tell from the seminar, this work that I'm going to describe was really based on... What's that? You can't hear me? Can you hear me now? So, just checking to see if people can hear me now. So you can, okay, great. Sorry for that. Just checking to make sure that it sounds like people are telling me they can hear me. Oh, great. So I just want to say, as you can tell from the seminar, there's been a lot of this work was really based on our involvement with state and local entities interested in doing this water reuse. So I was really very glad to be able to reach out to you guys and tell you about this work to a broader audience of the states. So as Jose mentioned, I guess I should start a little bit with my extraterrestrial background. I spent about 25 years. So we can, sorry, can you hear me?
Amy, can we check to see if others have audio?
Those online do, those calling in do not.
But Amy, you can hear me, right?
6 or call back in. Thank you.
All right, but okay, so let me continue with the talk. I'm assuming you guys can hear me. So I spent hold on a second.
The leader has unmuted your line.
Jay, are you online?
Yes, I am.
Okay, thank you. We can hear you now.
So, all right. I'm sorry for the technical difficulties. So, as I was saying earlier, and Jose mentioned that I spent a large part of my career working with NASA on these bioregenerative life support systems as depicted in these artist renditions of lunar bases or Mars bases or even floating space stations. And the idea here behind this work was to develop closed-loop life support systems where you're growing plants to provide food, water, oxygen, and recycling the materials needed to sustain life, so you could eliminate the need for resupply on these kind of longer-term missions. And I show these pictures here, which are of these artist renditions. More recently, there's been a movie about this. So now I can show you pictures of Matt Damon on the surface of Mars, a little bit higher tech photos of this kind of or media of this type of work. And I can tell you that I actually did grow potatoes, or we did grow potatoes on waste material, much like Matt did in the movie. So that's about the only thing I have in common with Matt. But the reason I bring this up is more about the design rules that this exercise or this experience taught me. And this quote here from Carl Schroeder talks about this idea that, if we could do this, and quite frankly, we can't do this right now. None of these systems exist beyond these artist renditions or media representations. But if we could do this, it means that you design human habitations independent of withdrawing or extracting resources kind of in a one-directional flow from the environment. Excuse me. So this concept, it's great to talk about this, waste as food and sustainable life support systems. And really the reason I came back to Earth, so to speak, from NASA was I really wanted to apply these kind of concepts to life on Earth. But it really does involve really rethinking and redesigning a lot of the types of our approaches to human habitation. In particular, one way to approach this where you kind of think about the problems that you have actually as a solution or a waste as more of a resource is when you think about buildings. And buildings, you can think about them as producing a lot of different kinds of water. It could be, whether it's water that falls on the precipitation on the roof of the buildings, or falls on grade or storm water around the building, or it might accumulate around the foundation of the building, or actually wastewater that's produced inside the building, be it source separated gray water or more of the black water and the toilet water, and even atmospheric condensate that's produced from the air conditioning systems. All those sources of waters could be viewed as a problem that you have to deal with and get rid of, but instead you could view them all as sources of water to reuse in the building, particularly not even for potable needs, but for non-potable needs, because the majority of water used in a typical building is for non-potable purposes, for flushing toilets, for irrigation, or other kind of cooling purposes in a building. So, and there are a lot of people, a lot of entities around the country that are doing this. This graph shows the Solaire building in Battery Park, New York City, that has been doing this for quite some time. They recycle 25,000 gallons per day, take the wastewater, run it through a membrane bioreactor treatment, and then use that wastewater or effluent from that reactor for toilet flushing and cooling and irrigation. And they've been operating that for over a decade now. And really their primary driver in New York City was not necessarily water shortage, but to reduce wastewater flow, because they have reduced taxes to pay on the sewer flow, which is of concern because of combined sewer overflow and pollution of the local waters. If you out west, there's a number of examples of these kind of building scale reuse systems. This is Salesforce Tower in San Francisco, which is a very large building. In fact, the second tallest building, I think, west of the Mississippi. And they're using, and this system's just coming online, they're using, again, a membrane bioreactor for treating the black water up to 30,000 gallons per day, and they again are applying it for toilet flushing, irrigation, and cooling, and that is really just starting right now. Now their drivers there, San Francisco itself, as the city is implementing and trying to promote this kind of non-potable reuses because of water scarcity, but they also were very interested in reaching LEED certification goals and also utilizing an existing dual plumbing ordinance that San Francisco has put in place. But it's not just I guess a coastal thing. This is an example from Minnesota, Lake Vermilion State Park, where they're the shower buildings depicted here, they're recycling the gray water that's produced in that building and reusing it for toilet flushing and saving, in this case, a little bit smaller operation, but up to 135,000 gallons per season. And their driver here, really because the drinking water was limited there, particularly because it's the naturally occurring arsenic that's in some of the local waters. And Minnesota also is involved in a number of projects. They have a very innovative stormwater and even melted snow system in a transit hub located adjoining Target Field that produces over 1 million gallons that's used for a energy recovery center. In this case, they're not using it inside buildings for toilet flushing or things, but they are reusing it for industrial applications. So there's a lot of this work going on, and about 4 years ago, I was involved in a group that met San Francisco Utilities, San Francisco Public Utility Commission. They led a meeting from a variety of states, and I'll show you a map next that kind of depicts where the people came from this across the country. Both public utilities and health agencies were participating, and the real goal was to look at this type of non-potable reuse in more the urban environments. And the this graph here kind of shows where people came to. The red lines show representatives from at the original meeting in 2014. This group has continued to meet, and I'll continue to talk about some of the outputs from this group. And the red asterisks actually show additional members that have been added to this over the last several years. And the red asterisk in the upper left-hand corner is for Alaska. Didn't have that on my map originally, but that's for Alaska. But that initial meeting, it was very clear that this group said there's two main things. We really want local management programs. They weren't necessarily looking for a national guidance on this, but they were interested in local programs, but they really were looking for what water quality parameters and monitoring were needed to protect public health, because these systems were either being permitted or they were getting a lot of requests for permits, and they wanted to ensure that this work was being done safely. So we developed a research program within ORD kind of related to this question of helping address the needs identified. Number one, if you look at these questions, how do you define the acceptable treatment? And also then how do you monitor that you're actually meeting that acceptable treatment? So those are the two big things I'm going to talk about today. The third question, though, is also about does it make sense to do this? And this really gets into some life cycle costing and impact assessment we've done at this trading, right? So if you're recycling water at a building scale, at what cost, right? What is the local cost of producing that water versus what cost you may be avoiding by reducing the amount of drinking water you have to produce and deliver to that site? But this graphic you see here on the right-hand side, it demonstrates some of the partners, the San Francisco Public Utility, San Francisco Water, Power, and Sewer, that Public Utility Commission. They really have been leaders in this. Paula Kehoe has been the chair of this group all through. And it started off at that one group I mentioned. Now there's the National Blue Ribbon Commission for on-site non-potable water reuse. And I'm going to be referring to documents that they've produced, guidance documents. The report that's shown was an intermediate stage. They had an expert panel that assembled, and they produced some recommendations. And now there's a recent document that came out in last December, really as a guidance for how to do this anywhere across the country. And the other parts about this, it shows that we're looking at trying to understand how to recycle rainwater, gray water, black water, or storm water at the local level. And on the left-hand side, it does indicate that we've tried to look at this question for different scales, not just the household level, but particularly buildings and large buildings that correlate to some of those buildings I showed earlier, as well as even districts of buildings or areas within cities interested in looking at non-potable reuse areas. But this question of does this make sense? Now, this is not a straightforward issue because it is complicated. There's lots of drivers for this. And this same group that I mentioned of the National Blue Ribbon Commission, again, representative of people from utilities and public health agencies across the country, one of the things they did was generate a list of why they were at the table. What were their interest in this type of reuse? Obviously, water scarcity and finding more water, in essence, by recycling it, is a big driver on this. But in addition, this non-potable reuse, there's a number of different efficiency issues that people are interested in. One is that you're kind of treating fit for purpose water. You're not trying to treat it all to a higher level for drinking water, but for the purpose that you need. And since a majority of the building water that's needed is non-potable, i.e. for toilet flushing or irrigation, there may be some efficiency by not having to treat water to as high a standard. Also, you're treating it close to the source and avoiding large purple pipe networks where you're transporting it over to a centralized system, treating it, and then sending it back out. And then also that this idea of having these more decentralized or on-site systems, it defers capital costs perhaps for large-scale infrastructure because you're taking the load off the need for, say, a large new water treatment plant. In addition, as I mentioned, like in New York City, another driver for this is not necessarily water scarcity and efficiently recycling water, but it's reducing pollution and loading to the sewers and the water bodies. That's also a driver in Minnesota because you're reducing stormwater flow. You're either reducing flow into the sewers and therefore risk of combined sewer overflows, but also reducing stormwater flows, like I mentioned at that Target Field example. And also there's the idea that this would increase resilience and adaptability of water and wastewater infrastructure because you're having multiple systems in place across a city. And if you talk to people like at San Francisco, there's interest in thinking about different nodes of within a city where you might have not just at a building or district, but maybe 10 or some number of recycling nodes across a large urban environment. And then finally, by doing this, it does generate some green space, particularly through irrigation in urban environments, which can be at a premium. And this would allow for meeting and exceeding green building goals, like I mentioned with the LEED, as well as, oops, sorry, got ahead of myself there. So there's a lot of drivers, but one thing we've done is, okay, if we go back to this question of, is it worth it? Let's just say if you take a building and you say it's 19 stories, 20,000 square foot, mixed use where there's some occupants in the building, also some business use, so, like some of the buildings I mentioned earlier, at about a 25,000 gallon per day wastewater. And if you're looking at wastewater or source separating the gray water from that building, and then treating it locally using some different approaches. And that bottom there talks about aerobic membrane bioreactor and anaerobic membrane bioreactor. Anaerobic systems may be a lower cost perhaps because you're not aerating them. They also allow you to generate biogas and recover some energy. Also, there's interest in green systems, again, to produce green space. There's technology called vertical flow wetlands, where it's kind of recirculating the wastewater through these wetland, kind of smaller scale systems that would fit in this kind of urban environment, as well as potentially heat recovery from this warm water that you are collecting locally. So we wanted to look at these different scenarios and say, okay, what's the cost of doing this at building versus what benefits there might be from avoided drinking water production or even energy that you might recover from either the anaerobic systems and the biogas that you generate locally or heat recovery from that. So this next graph, this next slide, it's probably the most, maybe the second most complicated slide I'll show today. But the simple way to look at this is that each one of those bars, you have the different technologies I mentioned, aerobic MBR, the anaerobic MBR, using a couple different operational parameters. One of the costs with this is the amount of sparging that you do to clean the reactor, clean the membranes in the reactors, the vertical flow wetland, and then an aerobic MBR that actually was trying to link to recovering the thermal energy locally. And you can see there that in one case, it's a natural, if you're replacing natural gas production of hot water versus electrical hot water production. So if you look at that graph, all those little slices represent different costs. The slices that are above zero, those are costs that you actually have to use to operate the system. And those are listed below, things like the cost of the biological process or UV disinfection, et cetera, right? It's listed there. The ones on the bottom are the costs that you're avoiding. In this case, the amount of energy you're saving by not producing the drinking water in the first place, or in some cases, those little hatched bars with the little diamonds on the bottom, those are energy that you're recovering, either from the anaerobic systems where you're actually using the biogas locally, or in the thermal recovery, you're actually taking that energy and using it, converting it into electrical energy using like a thermal heat pump. So in that case, you see where the dotted or where the clear dots are, the white dots. If they're below zero, that means that system is of net. It's a net benefit. It's a net positive type of energy transfer. If you not locally at the building scale, but overall for the city. So it may still cost to operate this building, but if you look at the benefit overall, it is of a benefit because you've avoided enough drinking water cost producing and distributing that water to the site, then more than you've actually spent treating that water locally. And so you can see there that most systems actually, the net benefits and are used for most options. There's some differences with the anaerobic MBR, for example. There's some higher costs associated with cleaning up the effluent a little bit more because it's an anaerobic process. Kind of gets into some details there. But the bottom line here is the overall point I want to get across, and it's kind of summarized in the next slide, is that if you look at the systems level, there's net benefits if you account for the avoided drinking water cost. And recovery of the thermal energy in particular can be particularly significant improvement, particularly if you're replacing if you're using electrical energy to produce your heat, hot water locally. Removing that is much more effective than trying to avoid natural gas, because if you're converting that thermal energy into electricity, natural gas just trades better overall. So, but there is some significant value in the thermal energy recovery, and like I mentioned, the system level cost of that chemical energy via the anaerobic bioreactor, it's diminished a little bit by the amount of energy that costs associated with trying to reduce the nitrogen from the produced water. And we're going to continue this work kind of looking at other options, including rainwater, stormwater, air conditioning, condensate, particularly as a function of different climates. So that's that question about, is this worth it? And like I mentioned, it's complicated. There's lots of drivers. And if you're interested in saving water, that itself may be of value. But if you look at cumulative energy use, there is some net savings there as well. So let's go back to the primary questions here. Number one is, how do you define acceptable treatment? If you're going to operate these things and you're going to permit them, how do you know that you're treating this water effectively so there's not additional risk, or at least acceptable level of risk by doing this? To address this question, I mean, one of the primary problems is that there are regulations or guidance for this, but they vary across different states. And this table shows you that for different states listed, here are different water quality parameters that they specify that the treated water should meet. And if you look at this highlighted area of where the bacteria, right, total coliform or E. coli, which are both indicators of fecal pollution, some states have levels for this, other states don't, and when they have levels, they vary. So that's the main problem. There's varying standards right now. And that was one of the drivers for that original meeting in 2014, they're trying to produce a standardized approach. On the bottom there, I want to point out that if you look at the typical gray water, the gray water that starts before any treatment, the levels can be very high, like 10 to the 7.2 means 10 to the 7th, right? That's to 10 to the 8th. We're going to talk some today about log reduction targets, as how much you have to reduce the load of microbes in these waters. And the reason you have to think about log reduction is that bacteria and viruses, the microorganisms that are in the wastewater, the ones of concern, are at very high levels. When you talk about microbes, you have to think logarithmically, right? So now in response to this varying level of from one state to another having different levels, different criteria, the National Sanitation Foundation, NSF, did produce this 350 water quality, which is standardized, which is definitely an improvement. And this does exist, but the problem with this approach is that it's not risk-based. And what I mean by that is that you can see that it does provide a single level of E. coli recommended for an average or for a single sample maximum. So there's defined levels and it's standardized. But the question is, what do those levels of E. coli mean in terms of risk? The fecal indicators are meant to be indicators of fecal pollution, and they are very useful in the environment when you're understanding, is a beach contaminated, for example, by fecal material. That is a good indicator of risk. But we know that feces are in this water, so in itself, it's not telling us what the risk is. To get at risk, you really need to adopt a broader framework. Number one, starting with having a risk-based target for acceptable level of risk. And this graph here shows that typically for drinking water, we assume that we want there to be no more than 10 to the minus fourth infections per person per year. If we're going to adopt this water recycling, a particular form of the non-potable reuse, we do not want to increase the number of infections by more than 1 in 10,000 per person, 1 in 10,000 people per year. Now that's for drinking water. The 10 to the minus second acceptable level of risk is actually the risk level used for recreational use of water, right? And partly you could ask, why do we have different levels? Well, one is that for drinking water, that's a requirement. You have to drink water. For recreational use, that is a choice. Those are also related to what we think the kind of acceptable in terms of what do we think the existing level of risk is. 10 to the minus fourth is kind of thought to be what the normal level of infection is from drinking water. So you don't want to do anything that increases it above the baseline. Now this approach is, the World Health Organization has used this risk-based target for wastewater reuse for agriculture, for example. The state of California is developing these kind of risk-based targets for direct potable reuse of wastewater into drinking water. So this risk-based approach is really the way the field is going in terms of water reuse. So this group that I mentioned really wanted to adopt this approach to extend beyond, say, NSF 350. And this graph here shows the kind of pictorially what quantitative microbial risk assessment, or QMRA, really involves. And this is the risk-based framework. If you look on the left-hand side, it really simply is saying, okay, it's going to take the different wastewaters, understand what pathogen reduction is necessary to meet acceptable level of non-potable reuse. And you need to, number one, understand what volume are people going to be exposed to when you flush toilets, when you use water inside, and there are some levels of exposure from that. You also need to understand what the density of pathogens are in that original water, and then also you need to understand the dose response of the pathogens you're concerned with in that particular water. So once you know all those things, you can predict what the risk would be if the water was untreated. Then you can say, okay, if I want to meet an acceptable risk, which would be either 10 to the minus fourth or 10 to the minus second, you define what level or log reduction you need to achieve to meet that acceptable risk. So that's really what the process is about. And if you fill this in with numbers, you set the problem, you understand what problem hazard you're interested in. This particularly is that you need to think about what reference pathogens you're concerned about for a particular water. And that's going to vary from one kind of water to another. For gray water and toilet water, you are interested in fecal pathogens because the feces are the risk. If you're thinking about rainwater from the roof, you're thinking about different kinds of pathogens, not human fecal pathogens, right? Because as I sometimes joke with these building operators, if you've got human fecal contamination on your roof, you probably have bigger problems to worry about than water reuse inside the building, right? The problem with collecting rainwater is not human fecal material, but zoonotic infections and wildlife use of the building. So you need to think about the organisms of concern, understand what the pathogen concentrations are in that particular wastewater, then understand what the removal would be for the primary treatment, disinfection, and then you have the non-potable exposure. What volume amount that you're going to be ingesting. This could be both the normal exposed amount when you flush toilets and just are handling the water, and also from accidental use of the water, particularly cross connections of water, try to understand what the rates of those are. So we built all of those exposures into our model, and then you also understand what the health effects are because you put in a dose response for the pathogens of concern, and that really then gets you at the risk characterization. And you, as that previous slide indicated, you define what treatment is necessary to meet an acceptable level of risk. And as I mentioned, reference pathogens, you need to understand viruses, bacteria, and parasites, all of would be concerned, particularly all of concern for wastewater. And then there would be different types of organisms, different types of bacteria and viruses of concern and parasites for, say, zoonotic risk versus human fecal risk. And then I just want to note real quickly here, and I know I'm probably running a little bit long too. So one of the key things is, what is that initial pathogen concentration? What we did with all this work is we go out initially and say, is there a number of studies, at least 15, using conventional methods that limit, that quantify pathogen levels above the detection limit, and then we would just characterize the density. But using that criteria, we really didn't find enough data. So in essence, you really have to model the estimates of the initial concentrations. And I'm going to talk a little bit about that in a second. So basically, limited availability of data on pathogen levels themselves. And that's one of the key things about this. I'm going to show you our results, and we've got log reduction targets. We also, there's clear needs for additional data to improve the models as well. And this is kind of the results. These are the levels of the log reduction targets necessary if you're going to use a given type of wastewater, which is on the left-hand side. Let's start up the upper left-hand corner. Domestic wastewater or black water, if you're going to use that for unrestricted irrigation or indoor use, either for toilet flushing or clothes washing, you need to develop a system that has at least an 8 log removal if you want to use the 10 to the minus 4th risk level, or in parentheses 6, which is if you want to use a lower list or higher acceptable risk of, then you only have to reduce it 6 logs. And that's for viruses, parasitic protozoa, and bacteria. So that defines what your system needs to operate at. All right? So that's really the basis of the results. We published this work in the peer-reviewed literature, and now this data has been translated. The table I'm showing you here is from the panel report that this National Blue Ribbon Commission developed, and which is now in the guidance documents that this group has released in December. So that provides some risk-based estimates of what treatment needs to do to meet acceptable risk. So just one, I think one of the things first people reaction to this kind of work is like, geez, you have to go through this model. You're making a lot of assumptions about the initial pathogen concentrations, what the exposure volumes are, and it is a modeling-based approach, and there is some uncertainties. For example, with the gray water and black water, if you just look at there isn't good pathogen data out there. So how we estimated what the initial pathogen concentration was we looked at the fecal contamination level in the water. There is lots of data on fecal contaminant levels in gray water or black water. We estimated what that was, but that doesn't tell you how many pathogens are in the water. The level of pathogens has to do with how many infected people are contributing to that wastewater at any time. So both the load of feces and particularly the load of users who are shedding pathogens at a time. So we modeled that by modeling what we think the frequency of infection is and the normal load of fecal matter is in these different wastewaters. And that actually gets you then the pathogen concentration. So this is what we did. This was our model, but one of the things you have to do is you have to go out and validate your model. And this graph shows that validation where we sampled, in this case, the SFPUC building, San Francisco Public Utility Commission building. We sampled that over a number of months in 2016. And the blue and green lines represent levels for norovirus, which is the virus of most concern because it's highly infectious and the one that you need to treat to treat the lowest levels. That was what we detected using qPCR, digital PCR. These are just molecular methods to detect virus levels in that wastewater. So the blue and green lines are our measured values. That solid black line is actually the level that the model estimated would be in water with the number of people that were in that building, because it does depend on the number of people in the building. If you have a huge population, like at a city level, you're going to typically have someone infected all the time. But in this case, smaller building, sometimes there's fewer, but when someone is infected, there's going to be less dilution of that. So, no, I'm talking a lot about feces here and pathogen things, but the point of this slide is to demonstrate that, yes, there are a lot of assumptions in the modeling, but then you go out and validate your model. At least for this particular aspect of the model, there's good agreement between modeled and measured values. So let's close out by talking about this third challenge or really the second overall question, but the third of the ones I've addressed is, once you defined your treatment system, how do you monitor that it's actually performing at that level that you've defined as necessary? Right? So the idea here, and this is what's again emphasized in this Blue Ribbon Commission report that I'm going to give you that's referenced at the end of the talk, is that you design a system to meet the risk-based performance targets. You've engineer has produced a system with sufficient barriers that it meets those log reduction targets. How do you verify that performance? Routine monitoring of indicator organs, like the fecal indicator organs, that doesn't really provide real-time risk-based information. As I've mentioned, it's not risk-based, and I'm going to show you some other data that just shows that even using those bacterial log reduction targets, typical indicator levels are too low. They're too low in the starting material, so they're not really the starting water, so it's really not going to be able to be a good monitoring tool. The proposed approach, what's emphasized in the Blue Ribbon Commission report, is that you really want to rely on operational monitoring, ongoing verification of system performance using some continuous observation. Could be like membrane, transmembrane pressure in a membrane bioreactor, the chlorine residual if you're using chlorine, UV fluence, things that you can really monitor online, because that's going to protect public health continuously. So those are surrogate parameters for the LRTs. That's really what's emphasized, to make sure that you're operating but reducing the sampling burden. But you still are going to have to, at commissioning, you're going to have to do some validation monitoring. And even when the system goes out of line, you're going to have to revalidate. You're actually going to have to say, yes, it's removing bacteria or viruses or parasites at those levels that we've defined. So what should that biological target be? The first response might be, well, measure the pathogens themselves. Well, the problem with that is there's lots of different potential pathogens, both bacteria, virus, and parasites, and within those categories, different forms of them. They occur sporadically, so you could be measuring it and it's zero, but that just means that your system may not be performing that well. It's just not being challenged at that time by someone who has shedding the pathogen. And it can be very expensive, and because very low levels are relevant, particularly for viruses, that the infectious dose can be just a few particles, a few units per mil, you can get a lot of negative results and spend a lot of effort trying to measure something and not really get much of a public health protection from that cost. The other option is to use biological surrogates that represent the pathogens. Typical surrogates like fecal indicators, I'm going to tell you they're too dilute. I'll show you some data to indicate that. The other option would be to spike with some surrogate and then calculate the reduction, like grow up a culture of bacteria or viruses, spike the system, and then look at the decay. That is done. That is possible. But that is a challenge to large systems because it's costly again. So the option that I want to talk about real briefly here is that we're looking at what we would call endogenous microbes as alternative biological surrogates. And these are organisms that are already there, not pathogens, not fecal indicator surrogates, but other things that are there at high numbers. And the reason why we're looking at this, and the overall approach is, number one, discover what those endogenous biological surrogates are. Understand what microbes are present using some high throughput sequencing technologies that are now available, which if you're not familiar with them, And you want to have more information on that, I'm glad to provide it. But the idea is there's a lot of work being done on, you've probably heard the term microbiome a lot lately. You're living in the age of the microbiome in the sense that the tools now are allowing us to see that we ourselves, you, everyone sitting there listening, you're more microbial cells than you are your own cells as you sit there, right? About a tenfold greater number of microbes in you and on you than your own cells. And we understand that now. That's always been the case, but we have the tools now to understand the level of microbial presence both on us and in the water and surfaces and the habitats we come in contact with. So that's where we're at now, looking at what those organisms are. Once you do that, you try to quantify them and look at what are abundant at a level above the log reduction target, so you can actually measure before and after treatment. These are things that are not harmful to you, but they're good surrogates for looking at performance. And then eventually you'd have to compare their log removal to the pathogens of concern and really develop these standard techniques or methods that you could use to monitor performance. So we're kind of in the middle of this zone now where we're kind of looking at what's there and quantifying their level. This graph here shows you some work with laundry water that we did. And the column just shows how many gene copies. These are all molecular-based techniques, again. And it's log 10 gene copies per 100 mils. And if you see along the bottom, the 4 columns on the left, those are all, well, the first thing I should show you is that this line here at 5, that is the level at which you need something to be above in the influent water to this treatment system to see our log removal, let's say, is target is 6. It needs to be above that because you want to measure before and after treatment to see you really got 5 log removal. So that's kind of the threshold. But if you look on the left-hand side, the typical fecal indicators, E. coli, Bacteroides, human-specific Bacteroides, or Enterococci, those are all lower than that level. But if you look at the skin microflora, they're at much higher levels. So they are above this dynamic range, which we define through the QMRA modeling. So that's one part of the story is, hey, looking at maybe more skin-associated organisms within gray water in particular. But that's the red that I mentioned there. But it gets even a little bit more complicated because the next graph I'm going to show you, and I know this is very complicated. This is a heat map of this deep sequencing we've done at gray water. So it allows you to look at all the different organisms that are in a mill of gray water. And it in this case, if you look on the left-hand side, that's a list of all the 30 most dominant species of bacteria we found. When we looked at the laundry, LA at the bottom, that is the laundry water itself directly out of a washing machine. If you look at that, you do see it's dominated by human-associated bacteria, human-associated skin bacteria, which is kind of what confirms what I just said before. Gray water, fresh gray water, is dominated by this human skin signal. But then when all the samples to the right, and I won't go into all the different classifications, but it's basically shower water that we collected after transfer through the piping systems in a building, at Colorado State University, where they were recycling shower water for toilet flush. If you looked at that, the human signal, there was some shower water samples where we saw some of that. You see in the kind of the middle there, there's a green. But mostly it was dominated by what are infrastructure organisms. It's the stuff that's growing in the pipes themselves. So by the time you're trying to treat the gray water, the things that are high level are not even the skin bacteria. But still, you want to get the log removal of the potential pathogens that are there. So maybe those are better targets, right? So even better surrogates from what I'd call the infrastructure microbiome. So just to kind of briefly summarize the monitoring work, and then I'll have one concluding slide. So for monitoring, the framework emphasizes online monitoring to best protect public health. So that's online, but quote unquote, we want offline biological measurements where you're actually taking a sample, going off and measuring it to validate performance at times. Typical surrogates like fecal indicators are limited because they're either too dilute or they might be the wrong target, right? So a human-specific fecal indicator is not going to be useful if you're looking at performance analysis of your rainwater, right? Because that's not the source that you're interested in. So what we're looking at is evaluating the microbiome to provide new surrogates, and we're working on both bacteria and viruses. I didn't show the virus work, but that's same kind of idea of discovering what's there at high levels. and eventually producing new standard methods to use for performance monitoring. And since these are the organisms that are at higher levels, there is a revolution in the area of sensors going on these days. So I think within the next decade or so, we will have online biological sensors, but you're going to have to target the things, you're going to be better off trying to target the things that are higher concentration, like some of these surrogates that I've mentioned. So just overall summary, in terms of impact of this work, what I wanted to try to get across is that the immediate impact is that because we worked with the states and asked them, what do you really need here? What they wanted was tell us how we should define the necessary treatment for these systems. The work that we developed, the peer-reviewed work, the log reduction targets, has been directly incorporated in this guidebook for developing and implementing regulations for on-site non-potable water reuse. And that's online. There's a website in the final slide that you can go to too, which has all the resources from this National Blue Ribbon Commission. So in essence, we're providing these public health agencies direct guidance on treatment. And then I think the ongoing work is defining more effective biological targets for monitoring performance and developing associated standard methods, as well as the initial things I talked about by looking at the cost-benefit analysis of different non-potable reuse approaches to inform best designs, both in terms of the scale of the treatment systems as well as the type of treatment systems. And just to conclude, these are the additional resources, both from the U.S. Water Alliance, which is the documents from the Blue Ribbon Commission. I also want to mention that if you want a little bit deeper dive into some of these details, I will be giving a seminar through our SSWR, our Safe and Sustainable Water Resources Program, on Halloween to talk in more detail. And also, this is going to involve a panel discussion with commission members from both two different utilities as well as two different public health commissions to kind of talk about where the status of this is in their local area. And then finally, at the end there, I'm talking here about a lot of work, but I'm really do most of the talking. Those people there, particularly people in that first line, Nicole Brinkman, Scott Keeley, Michael Jane, they were really responsible for doing the majority of the work that I described. So thank you. I'm glad to answer any questions.
Thanks very much, Jay. So if you have questions, please